Recording in progress.
Good morning, everyone. Today's topic of discussion is functions and turnover of cell wall. So far, I have discussed the structure of the cell wall, various components of cell wall. Now coming to its functions. Cell wall accounts for characteristic shape of the cell. It's a structure with high tensile strength, so it adds to rigidity of the cell and provides mechanical support to the cell. It has important role in cell division and it forms cross walls to separate daughter cells. So, new cell formation, cell division. Cell wall contains antigens important for virulence and immunity. This is a very important function of cell wall for protection of the bacterial cell in so many aspects: shape, strength, barrier, separation of daughter cells, that is new cell formation or cell division, and virulence and immunity. It helps transport through porine channels and also some exert resistance to antibiotics by modification of porine channels. If porine channels don't allow any molecule, so the antibiotic molecule, then the bacterial cell will be resistant to that. Some shows antibiotic resistance by alteration of penicillin binding protein on cell wall. All are the functions of cell wall, different functions, but the most important function is that it prevents osmotic burst or lysis of the bacterial cell. It supports the weak cytoplasmic membrane against high internal osmotic pressure of the protoplasm. When the bacterial cell is inside human body, suppose it is in the blood, then there are huge osmotic pressure difference between the inside of the cell wall and outside of the cell wall, but and there is always a chance of osmotic burst or lysis of the bacterial cell, but the cell wall prevents such lysis or destruction of the bacterial cells. This is the most important function of the cell wall. Now coming to the turnover of the cell wall. There is always lysis of one layer and then synthesis of a new layer. So the cell wall biosynthesis is a continuous process. Bacteria themselves possess enzymes known as autolysins which are able to hydrolyze their own cell wall. They can synthesize new polymers which are added to them. That's why bacterial cell can grow and divide. So new peptidoglycan polymers are exported from the cell and gets linked to the already existing cell wall polymers at the inner aspect of the cell wall by penicillin binding protein. At the same time, older peptidoglycan structure overlying the newly synthesized structure gets continually removed by autolysin. Just like peeling of onion from outside inwards, the outer layer being peeled off and the inner layer, that is a new one, and it's for cell wall the newer layer gets synthesized from the inner side and then gets attached to the cell, to the already existing cell wall polymers. It gets linked at the inner aspect. Similarly, like autolysin, lysozyme is an enzyme which is normally present in tissue fluid, which lyses susceptible bacteria by splitting the cell wall mucopeptide linkages between NAG and NAM molecule, which we have already learned. So lysozyme splits the linkage what is present between NAG and NAM molecule, that is the beta 1,4 glycosidic linkage. So it can completely destroy the cell wall of gram-positive bacteria. Thus, what is the importance, what is the application of knowing this turnover and function of the bacterial cell wall? Because knowing that we can attack the bacterial cell by targeting its synthesis and or its structure and penicillin binding protein while designing antimicrobial agents. Those are made in such a way that it can stop its synthesis or it can disrupt its structure or disrupt the penicillin binding protein. because the cell wall is very much essential for survival of bacteria. Once it gets disrupted by antimicrobial agents, then the bacterial cell will be destroyed. Now, synthesis of outer membrane protein. These are synthesized on ribosomes present in the cytoplasm, which are bound to the cytoplasmic surface of the cell membrane. But how they get transferred to the outer membrane proteins, how they get transferred is not still clear. One hypothesis is there, that is, this transfer occurs at the zones of adhesion between cytoplasmic membrane and the outer membrane. That means the ribosome present on the inner surface of the cytoplasmic membrane, they synthesize the outer membrane protein, and then they are transferred to the zone of adhesion between the cytoplasmic membrane and the outer membrane. This adhesion is visible on electron microscopy. This is the intermembrane space, this is the inner membrane, and this is the outer membrane. So at the junction, these get transferred. So that's all for functions and turnover of cell wall. Hope this can help you to make notes and for better understanding. So thank you. Remember to like, subscribe and share.